So taking us in this session is Seun Olufemi, Michael Olufemi, and Adedoyin Adesoye. Our speakers ready? Our speakers ready and present. Adedoyin, Seun Olufemi, and Michael. I can see them in the house, and are you ready to take over now? Israel, yes, you can. The recorded video will be posted sometimes later, and once it's posted, the information will be passed across. Oh, okay. Just to start the flow, I don't think I introduced myself. Okay, my name is Emmanuel. I'm a member of the Science Cafe Chaperone team, and I am officially welcoming us once again because I just remember that we have some new members, and I probably want to know who is speaking. Yeah. So welcome to tonight's session, and like I said earlier, let us be ready to get as much information from this session as possible. And as many questions you would also have from the session, please put them down in the chat box. Going through the session, please let there be no interruptions except the speaker points out. And please let us be as engaging as possible. When there's need for comments, let us please drop comments. When there's need for questions, let us drop questions. And then please let's not interrupt the sessions. Let's keep our mics muted until it's time to speak. And then once it's time to speak to, please raise your hand and you'll be pointed. If not, drop your questions in the chat box and your questions will be read out. Thank you very much. So I have been told the speakers are ready and on ground. So I would say over to you.
Hello everyone, can you hear me?
Hi Sean, yes, I can hear you.
Yeah, good evening everyone and welcome to this special edition. So it is our joy and our delight to teach members of the community, and special thanks goes to the coordinators. So am I permitted to share my slide?
Yes, Sean, you can. Yes, you are.
Okay. Please, can you all see my slide?
No, not yet. That's not shown yet.
You should have. Can you see my slide now? Can you see my slide?
Yes, I can.
Yeah.
Okay. So, welcome. So we'll be taking through basic bioinformatics, and I'm privileged to teach with some big guys on bioinformatics, and Mr. Michael and Ms. Adedoyin will be taking the section for Wednesday through Saturday. So my aspect of this training will be basic bioinformatics and biological databases. So I'll try to, actually this is 2 modules, but I'll try to be as fast, apt, and possible. And we're going to be having some tasks to do. We'll be having taxes to do. I hope our systems are ready with us. And then we'll also be having a brief section on what bioinformatics is. So I am Sheon, a student of biochemistry, Ladika Kantala University of Technology, right here in Gomosha, Nigeria, for those who doesn't know. And I'm also a research assistant at Elix Belgian Institute. I'm interested in molecular biology and how we can use bioinformatics and other molecular tools to solve current problems. So I will consider this baby or this cartoon as Siri or whatever the name we might give to it. Okay. Now, he's asking a question for the newbies amongst us. What is bioinformatics? So I'm the one answering now. Bioinformatics is an application of information technology to process, store, organize, and analyze data generated in biological research and experiments. So here I highlighted information technology, analyze, data generated, biological researches, and experiments. So it's as well, bioinformatics and computational biology are well rated in life sciences as well as in computational, computer and information sciences. Majorly, it is the application of computer-based algorithms to analyze biological data. Now, here's bioinformatics. Here are the fields that are interrelated with bioinformatics. We have biology, computer science, information engineering, mathematics, statistics. So the word bioinformatics is just majorly two words. We have biology and we have informatics. Biology deals with our general body, environment, study of living things, body, environment. I hope you can hear me.
Yes, loud and clear.
Yes, I can hear
Okay.
you.
So body environment, while informatics on its own has to do with the information technology aspect, statistics, the data analysis aspect of it. So we can divide the word bioinformatics into two: data generation and data analysis. So the biology does the data generation, while the informatics does the data analysis. So
Hi, Sean.
now,
Sorry, please. Hello, Sean. Please, could
yeah.
you press the slideshow option to make the presentation bolder? Because it's seeming complete.
Okay.
The one close to the share option, that slideshow button. No, slideshow close to the share button at the right side of your screen or left.
Okay, yeah.
Yeah,
Hope it's clear now.
yeah, it is.
Okay. Now, the baby is also asking me a question again. The newbie is also asking me a question again. How do bioinformatics come to be? So, generally, the study emanates from classical genetics, right from the discovery of DNA, and gradually to we have some notable things we could talk of. 1968, we have the day of the kind of they are the first to create a protein sequence database. In 1970, we have sequence alignment algorithm, 19, 1970s, 1977s, and all that. Then down to 1990, that is when bioinformatics work started getting really, really appreciated when we have the Human Genome Project. Now, it's also asking me another question. Okay, who can study bioinformatics? Generally, bioinformatics can be studied by all life scientists, programmers, statisticians, data scientists, and all. So anybody whose its study involves the environment, biomolecules, DNA, RNA, protein, signaling networks, cell organisms, and the human body can study bioinformatics. What does bioinformatics has to offer? So here I want to talk about the aims of bioinformatics. One, the aim of bioinformatics is to organize data in such a way that it allows researchers to access existing information, to submit new entries that are produced, to develop tools and resources that aids analysis management of data, then to analyze. Now, majorly, it helps researchers in the pharmaceutical industry, so they move from trial and error methods down to computer-based approach for designing drug, which has so far helped in the process of drugs and others. So, another question the newbie asked me is, what do I need to become a bioinformatician? Because he's interested in becoming one. So, what does the newbie need? One, a working personal system. And I meant a working personal system. An internet source, because you'll be needing that. A strong knowledge of molecular biology and field of interest. Molecular biology is general because he has to understand what's the server's algorithms. He has to understand molecular biology to be able to interpret. The reason for this is Bioinformatics helps you in analysis. It doesn't help you in interpretation. So your molecular biology knowledge and your field of interest, be it immunology, be it microbiology, be it food, be it virology, or anything, or parasitology, or whatever field of interest you are interested in, it is the knowledge that you will use to interpret the results you have. Then these ones that I bullet, storage system, programming skills, and statistical skills, are the ones the person will need while moving on. Storage system slash cloud computing, that is when you want to analyze a very large data set. Programming skill, you'll discover that as you move on, you would need to learn some few programming skills, Python or R. Then statistical skills. Then what does a bioinformatician do? So after the newbie has understand what bioinformatics actually can do, so he wants to know what bioinformatics do, like what is their choice. One, they analyze data interpretation, they create databases, and they develop algorithm and statistical methods. So majorly that's what a bioinformatician do. What does it cover and where does it apply? Bioinformatics is widely used in the area of medicine, microbial genomics, and agriculture. So below I list some applications of bioinformatics in life sciences. One, in drug discovery. This work here was done by the PA of a institution in conjunction with some others. So this was a computational study that checks the effect of cola as an inhibitory potential on COVID-19 main protease. So all this kind of works, when put together to progress, it helps in drug discovery. And instead of the normal pharmaceutical trial error method, whereby you have to go to the lab, start screening your leads, check for bioactive compounds and the likes. So you just do bioinformatics, and since we already know some of them, then we move. Now, in molecular phylogenies, this one you will be taught towards the end of these sections. That is phylogenetics. So this is phylogenetics and phyloevolutionary analysis of SARS-CoV-2 in Nigeria. So this study was done also by my Oga and some other people. Now, what they do is actually they check the evolution. This is when COVID just started. They check the evolutionary traces of COVID in throughout Nigeria to check the relatedness and the likes. So they check the molecular conditions and how they do. New emerging aspect of bioinformatics that is really, really moving ahead now. So under immunoinformatics, we have a lot, and this is where my institute actually specialize. Worked on vaccine design, work on point of care diagnostic kits, in vitro peptides, um, mRNA vaccines, therapeutics, RNA therapeutics, and the likes. So instead of COVID as a whole, well, gave us an edge. This work actually is already moving ahead and is moving to its clinical stage. So they designed a conserved peptide-based subunit vaccine for SARS-CoV-2 using immunoinformatics approach. Genetic epidemiology. Genetic epidemiology is one of the most interesting aspects. So it checks genetic variances between genes in a large data set. So if you are wondering that, okay, if I study bioinformatics, I may not be able to do something new or to discover new things. That's the reason why I put these here. New discoveries, variants. Omicron was discovered by an African scientist, Tolu de Olivere, and he discovered it using bioinformatic tools, a particular regression model. I cannot remember per se. So, new discoveries, variants. Oh, sorry. Then another one, new discoveries. This work was done by Dr. Shegun Fatuma, where they actually did a form of genetic epidemiology, and they found out that it is possible that some of the diagnostic tools or diagnostic biomarkers we have been using to diagnose for some diseases generally might not work for African population. So this work is still under view. I mean, it's still under some view. So this work is actually telling some functions of some genes in the human body that we may not be actually be taking proper cognizance of. So are there any career prospects in bioinformatics? So I listed some job works here: bioinformatics computer specialist, analyst, programmer, analyst, scientist, consultant. So as you can know, bioinformatics is widely paid. So you can get your job in research institute as a data scientist and the likes. So also, yeah, the field of bioinformatics could be applied in genomics, the study of the whole body genomes, in metagenomics, in transcriptomics, metabolomics, cellinomics. Okay, so the field of bioinformatics is ever trending. At this junction, before we move to our first task, do we have any questions? Do we have any questions before we move to the second module? Hello, is someone with me?
Yes, we can hear you. Please, if you have questions, please drop them in the chat box and I'll read them out for you. Or you could raise your
Okay.
hand and I would point you out. But for
Before
the
we move to this,
so far, Shimi, there's
before
no.
we move to the second module.
Okay. I've seen I think there's no question yet.
Okay, thank you. Now, here is the tax one for my training aspects. The tax one says you should name your research interests or field of interest slash course of study. Then, how can bioinformatics help you become better at what you do or what you will eventually do? Then the second one says, do you have a specific research questions you can please share? So this particular task is to be done and submitted to Sheunlufemi123 at gmail.com. I would drop that now. I will drop that now. Okay. Yeah. Thank you very much, Mr. Ikun. Shame Olufemi123 at gmail.com. So that is the first aspect. Now let's move to the real work here. Introduction to databases. So what are databases? Well, I will not say database. I'll call them biological databases. So biological databases are repositories where biological data are generated from biological experiments and they are stored, assessed, organized, and are managed. So the contents of biological data such as protein sequence, molecular structures, DNA sequence in an organized form. Now, they allows our biological data to be traceable as they are indexed using accession numbers, and they also contains preliminary information about the data, which is called metadata. I will show you this one by the time we get there. So what are the features of a biological database? Number one is they are complex. Two, they are heterogeneous. Three, they are dynamic. Four, they are inconsistent. Five, it contains high volume of data. Five, another one is data sharing. Then it is highly curated. So here's a question. Why do I even need, as a bioinformatician, why do I need to access sequence status? Why would I want to access sequence data? Number one of it is to know the sequence of a gene. So if I want to know the sequence of a gene, the structure of a gene, if I want to check out um, the variance of it, the variance in a sequence, if I want to compare my sequences with others, if I want to identify, there's what we call blasting, if I want to check the work I've done, has it been done by somebody or was it related to? So then if I want to find the diseases in association with the my variation of interest, so I can decide to check for that. Another question is, what are the objectives of the biological database? One is to make data available. So when you're checking biological databases, is to make data available. Two is to store relevant information easily. Another one is to make biological data available to scientists to work. The first one says available and to make it work at a place. Then so that we can easily get updates on existing information easily. So uses, so it helps us to easily make UTSs, antivirals, identify useful helpful bacteria. Um, it acts as a storage of information. It helps remove redundancy. Redundancy in the sense that, okay, for example, somebody within your community has isolated a particular strain of a virus and has submitted to the database. So it would help you know, okay, that this part of work somebody has done by the time you're searching your databases. So the types of database, biological database we have based on the nature of data, on structures, we have PDB, scope, CATTH, on proteins, Swiss proteins, tremble, prosites, nucleic acid, gene bank, DDBJ, um, EBI. Then based on sources, we have primary, secondary, and we also have specialized databases. Let me say that. Specialized databases are databases that are specialized particularly for probably an organism. Like we have the GSID specialized for respiratory organisms. We have the Viper, and so these are some of the most popular databases we have. Uniprot, ProSite, Faster, Woo, Embers, Sprints, Scope, Blast, Swiss Model, Sanga. So, for the sake of this lecture as an introductory class, I'll be talking on primary databases alone. So now, primary databases, they contains original form of information, and majorly what we try to do is to check for the nucleic acids, sequence and protein sequences we have in them. DDBJ, EBI, NCBI. Now, okay, I'll come back to this. When you try to access NCBI, this is the interface we will have. So I'm still coming back to this. Here's the interface we will have. Here's the sign of it. For EBI, this is the interface we will have. For DDBG, this is the interface we will have. Now, there is an agreement signed by these three primary databases which make them collide to form the INSDC. And the major thing in the agreement is that any sequences you submit to either of these databases is reshared amongst them, but their session numbers or their index numbers may be different. So if I submit to NCBI, so within a specified time, my sequences and the informations I provide on NCBI will also be at ABI, will also be at DDJ. And that's how they rotate. So necessarily, if I'm getting my data from EBI, I do not need to go and check again from NCBI because they regenerate data. And that is quite important for you to know as a bioinformatician or bioinformatics enthusiast, that in order to reduce redundancy in your work, so that you not just be having the same data coming over and over and over again. When I was talking about metadata and when I was talking about database data time, I mentioned two important things which I will show to us at our aunts on laptop section. I mentioned the metadata. I also mentioned add session number or index numbers. So now, yes, hands on laptop, our section. So now you're about becoming um, a bioinformatician. Are we there? Do you want to say
Yeah.
anything before we move?
No, there's been no comments yet. You can go
Okay,
on.
yeah. I hope everyone is with his or laptop. Okay, so I will advise you to use Google Chrome or Brave. If you are using if you are using Microsoft Bing, I think you might be giving us a little problem because sooner or later you discover that that particular browser will be giving you issues using bioinformatics. So Google Chrome or Brave will do well. So go to your Google page, open a new tab, then type NTB NTBI. So you search. Are we there? Please, how many of us didn't get this page?
Yeah, we're with you.
Yeah. Okay. Now, I think there's something I'm forgotten now, but now I just remember to say it now. When accessing database, we can use two approach. The normal one approach or the layer approach or the layman approach. And two, you can also use command line to access databases. Then you'll be using virtual machines and using virtual machines and Ubuntu to do that. But for this section, we'll be using the normal approach. Now, it also skipped my mind to mention that NCBI is sponsored by NIH, the National Institute of Health, USA. EBI is sponsored by the molecular laboratory at the Europe, United Kingdom, while the other ones were sponsored by Japan. Now, something you should note about NCBI. So majorly what we will do now is we will do sequence retrieval. Mr. Olufemi will come and teach you how to do genome assembly, but what I will just put you through is sequence retrieval. But just get this straight, or get this. NCBI is not only a database, it houses many other databases. So you can just come to NCBI to do some of your works. So it has a lot of things that you can do with it. It has a toolkit in it, incorporated in it. That's one. Two,
Please, sorry. How
NCBI,
did you get to this page again? Very sorry. How did you get to this page again? Sorry.
yeah, you come to your Google Chrome, open a new tab. Are we there?
Yes, we are here.
Then type NCBI. Are we there? So this is the interface. It's just a so this is the interface of NCBI. Okay. Now, as I was saying, navigating through NCBI. Now, let me show you some of the things we can do here. The popular PubMed, where you can retrieve, where you can use to get papers, is also incorporated in NCBI. Here is PubMed. Here is PubMed. So if I want to go to PubMed now, I can just click on PubMed here, then search the name of an organism or anything I'm interested in also. So it contains quite a lot of things to do. I can search for a gene, search for clinical variants. Even I can use it to do conserved domains, to check for my protein clusters, bioacids. For the people that will be using, for people in computational biologies that works with PubChem to retrieve structures, small cannolis of compounds and the likes. So NCBI is still their primary home. So I've established the fact that NCBI is an home for many or numerous databases, one. Two, it also contains a toolkit whereby you can access or do some of your works. Now, please everybody go to nucleotides. Nucleotides. Are we there? So you press enter. So now, I would want us to suggest the name of an organism you want us to work with, to just retrieve. Okay,
E.
thank you,
coli.
Rotimi. E. coli. Okay, somebody said E. coli. Sorry, E. coli. Now, coming down to, remember, before I start here, I changed my database to what? Nucleotide, Abby. Now, this shows that everything I'll be seeing on this particular page will be based on what they have in their nucleotides. Had it been I changed this to, okay, general, like just put that all databases. So I just want to see a combination of what they have on probably their PubChem, PubMed, and everything. So I will see quite a lot of things. So I have 33 results in I have 33 results in I have results in 33 databases. So now come to see in Bookshelf, I have over 6,000 books written on it. In PubMed alone, I have over 430 something thousand on it. In PubMed Central, over 600,000. So in nucleotide, I have over 19 million. So in protein, I have over 71 million. So this might actually be too cumbersome for me to work with. So it is advisable for you to just come to the particular database you want to work with. Then you streamline it down, nucleotide, then you search. Now, imagine on E. coli. Hello?
Yeah, sure, we can
On
hear
E.
you.
Yes, on E. coli, I have over 19 million. Probably that's too big for me to work with. It's too much. But we have over 19 million sequences. And the very first one now is even a protein. It's a protein. So coming down. Now, please note, I don't remember to include it in my data, in my notes. Databases are not humans. They don't think. So they only work based on algorithms. So the fact that you search something on a database and you couldn't find it does not mean that it does not exist in a database. Are we getting that? Hello, are we getting that?
Yes, yes, we are.
Okay, please drop
Okay. So,
responses on the chat session just to make sure know what we can hear him.
okay.
Because you have the chat session as engaging as possible. Thank you.
Okay. Now, so for E. coli, I can say now I want to work with E. coli, but what I'm interested in working with on E. coli is sorry. Let me see the partial sequences we have. Sorry. Now, I said partial, Abby. Can anybody see what I wrote here? So the fact that it is wrong makes the database to tell me that there's nothing that exists like that, no item found. Now, imagine when I change it to partial. So can you say I have something? So, so in assessing database, it is quite important for you to know that your query statement is quite important. Do you understand that? So here I have a lot a lot, a lot, a lot, a lot to work with. Now, let me introduce a few things to you. My time is almost up. I only have 4 minutes left. Let me quickly introduce some things to you. Now, clicking on the very first one, can you see now? I pressed E. coli. What it brought out was not E. coli, okay? But due to the fact that some of my search queries matches it brought it out. So it works the same way just as Google, whereby sometimes you search some things and what you have might be different. So you should be wary of that. That's the reason why I stated earlier that a database is inconsistent. Now let's just quickly check this. When you come under here, I hope I can. Okay. Yes. Now the accession number I mentioned the other time. Can we see it? I need to know if people are getting me. Can we see it?
Yeah, they are dropping
Yes, we can.
responses. Yes, we can.
Okay. Yes. Now this accession number shows it's just like when you get admission to secondary school, I mean to a university, you're given a particular number that belongs to only you. So this particular number in this database belongs to this organism alone. And interestingly, you can also use it to search for it. It's just like when you want to enter an exam and they call your matric number to come and have the front seats. So so that's the way it is. Now here's GenBank. Here is faster. Here is graphics. Now I mentioned metadata the other time. Okay, I mentioned metadata. Now, using some other specialized database, if you are working with something like GIS8 or the likes, you may not have it as straightforward as this. So, whereby you just have to match some different compartments together. But for NCBI, so what you come is you come and click on GenBank. So when I click on GenBank, remember this is E. coli, the strain number. So this is just an RNA gene, a partial sequence. The GenBank here would give you every information they have about the organism or about this sequence. So it gives the locus, the accession number is given. So it gives the organism the source. It gives the name of the author, how it was submitted, and every process that was going through it. Now, let me show you something quickly before we talk about some other things we could do here. Now, one of the things I said earlier that you should have a strong knowledge of molecular biology is this. What I input in was whole genome sequence, but here in my structures, I'm also having what's contigs, okay? And assembly contigs also indicates fragments. So let's quickly assess this. Let's assess this. I want to show you some things. So I think what I'm downloading is a very large data sequence, but let me just, okay, yeah, it's done. Now, when I am assessing a whole genome sequence, can you see CDS gene, CDS genes? So they will show you different things on it. Each genes you can find, you can even get their numbers, their ID numbers, where you can read more on them if probably you want to work with them. CDS genes. Then gradually, by the time you move down, so you see sequences of those genes. Then before you come down with this. Okay. Now, let me show you one finite thing here before I show you how you can retrieve your sequence. Okay. Can we all see this aspect? Where my sequence is, where my cursor is. Can we?
Because
Hello?
customize.
Yeah, this aspect.
Yes, you can.
Now, moving down, this aspect shows some information pertaining to the sequence you have downloaded. Okay. So here there is something you can do. It all depends on your knowledge of the course or the knowledge of the sequences you want to retrieve in. Now, if, for example, I know that, okay, maybe E. coli, the whole genome of an E. coli, or the sequences I want to work with, probably is a particular gene I want to work with. Let's say, for example, I want to work with BRCA2 genes, okay? Now, and I know that BRCA2 genes, the base length does not exceed, let's say, let's just say 1,000. So, and I know that does not exceed 1,000. So I can just come here to the sequence length and type 1,000. Let's say maybe 800. Then here I can say 1,000. So all the sequences I want to appear is between 800 and 1,000. So what it will do, I'm not sure we can get something of that. What it will do is that it will filter everything that is not within that range. Can you see? 829, 921, 333. Do we understand that? Okay. Now, I've exceeded my time. I'm very sorry to the next trainer. I've exceeded my time. I've exceeded my time. I'm supposed to round up now. But let me just show you how we can retrieve sequence, and that will be all. So the other two primary databases that I showed us, um, EBI and DDJ, you can just go and navigate around it and see some other things. So this database you see, and actually there's a tax that is attached to this, so you'll be able to work with it. Now, how do I retrieve my sequence? Okay, imagine a question that, okay, I'm to retrieve a sequence that contains over 200, I have to retrieve over 200 sequence. How would I do it? Now, when I click faster, normally we click, normally sequences are stored in databases in faster formats. We have the SRA, we have, and some different other SRA, faster, FASTA, and different things like that. Now, the faster sign, here is the faster sign, greater than. So, in other bioinformatics In other bioinformatics training that you'll be doing or that you'll be moving on to, in other bioinformatics training that you'll be doing or you'll be moving on to, so it is very much possible you would see that you have to submit your sequences in faster format. Are you there? So here it is. So by the time you're submitting in faster format, so note at times the sequences you retrieve may not actually be in faster format. So you will just have to include this sign by yourself. And some might even ask you to save in .faster. Now, coming down to this is an example. I have my faster sign, then here is my sequence. Here is my section number. Sorry, here's my section number, my faster sign, my section number, and this is just a content. So how do I do it? Well, some people will just advise you, well, if I'm not working with too many sequences, I can just decide to copy. I can copy singly like this. Then I press Ctrl C. Is somebody doing that? Then I come here. And I paste. Please note, when we are working with sequences, it's not advisable for you to use Word. You can use your notepad or your word pad because because of the mode to save. Sure you get. So if I copy this sequence now, then I can come in with here, I can come file, then I'll save as. So I'll save it to a place where I know I can easily use it. Let me save it to my desktop page. Then I can save it with SC lens bioinformatics. Okay. So once I click on save, it gets saved. So I already have this on my system. Then from immediately I have a sequence, then I can do a lot of things with it. Everything I've mentioned about in the application, I can proceed to move this to informatics. I can even model the sequence. I can take it on to start checking for variant calling. I can do a lot of things. I can even do NGS analysis. I can do comparative study with it. So this is me just retrieving just one sequence. Okay, now, coming back, what if I want to retrieve, let's say 20 sequences? So what do? I can easily just come here and mark. I won't be doing this because we are in training section, because I'm not working on it. And remember, it is possible you have an intruder in between them. So try to check in working with sequences or working with databases. You have to be very, very vigilant because it's possible you have an intruder within them. So you just mark, okay, let me say I've marked 7 now. So what do? I come to summary. So clicking on summary, so I can click on this just faster. I can download everything as faster, and I can download it as faster text. Let me say faster now. So faster text would bring everything for you on a singular page. In here, you still have to copy and paste. But I don't want to copy and paste, just like I've did the other time. I want the system to complete everything for me straight. So since I've marked it, so back here, so I just come to click on faster. So once I'm done with that, I click on send to file. So then I clicked on it's going to download it in faster format, then create file. So since, okay. So that's how it's going to download. Now, I mentioned metadata. Probably if I want to work on this and I want to do something like genetic epidemiology, I want to check some um, particular informations across the territory, across places, across people, age groups. So I will have to need information as pertaining to each data. So what do I do? So I can download the entire information alongside with it. So how do I do that? Still coming to my summary, yeah. Then coming to my summary, I can click on GenBank. If I click on GenBank full, it's going to bring it on that same page. Then I'll do what? Come to my send to. Sorry. So when I click on my GenBank, it brings everything out in each one. So I just have to be clicking on it one after the other. Okay. One after the other. So if I change it to GemBank full, now this GemBank alone would hide some informations, but GemBank full brings out all. So I wait for it to load. Then I choose my file. Sorry. So I can download. So make like create file. It's going to start downloading too. So it's going to start downloading. So likewise, if I download the Gen Bank, likewise, if I do this and then download the Gen Bank alone. So it's just the same procedure. Until I choose my file, it comes down, then I can create my file. It's downloading. So I hope we're all getting it. Are we getting it? Do anyone has a problem? Hello?
We're following you.
Yeah, do anyone has a problem? Okay. So let me just show you EBI platform. We will not be working with that.
Sorry to interrupt, but please, we are running out of time for this session. So if you can start wrapping up for the next
Okay.
speaker to come up.
Yeah, thank you very much. So here's just the EBI platform. So you can do similar things with that. Okay. Um, so do we have any questions? If no questions, here's our second task. In order to do that, we have to retrieve at least 10 whole genome sequence of E. coli, stating their location in FASTA format from NCBI. You are to retrieve at least 10 whole genome sequence of S. aureus with their metadata. So then this assignment has to be submitted to shinwolufemi123@gmail.com as earlier stated. So I appreciate the management of Science Cafe for inviting me to come and teach other community members, Bioinformatics. And I also appreciate my principal institutions and Elix Margin Institute. I also appreciate every organization that has agreed that for me to learn with them in bioinformatics and other molecular fields. Thank you very much. Thanks for listening and thanks for coming.
Thank you very much, Sean. Thank you very much for the session. We can see our next speaker is on ground. So please, I'm sure most of you have questions. Let the questions be dropped in the chat session, and then even if they're not attended to today, they'll definitely be attended. So let us put down our questions. But in the meantime, thank you very much for your active participation. Thank you, everyone. So hello, Michael. I see you're ready to take us on the next session. Please, you can go ahead.
Good evening, everyone.
Good evening, Michael.
Yeah, so can you hear me?
Yes, I can hear you.
Okay, yeah, thank you. I will just go ahead and share my screen so I can start right away.
Okay.
Is my screen visible to all?
Yes, it is.
Okay, thank you very much. So good evening, everybody. I'm happy to be here to participate in this workshop and introduction to bioinformatics session. So my name is Michael Olufemi, and I'll be taking you guys through genome assembly. So I'll try to be as fast as possible so you can see some practical hands-on. Okay, so I'm starting with what genomics is. So genomics itself is the comprehensive study of an organism's genome with a focus on characterizing them. And as the genome itself is the complete set of an organism genetic material, that's that we understand that what we use genetics to study an individual gene. But when we talk about what genomics, talking about studying the complete organism's genome and not just individual gene. And what a gene is a portion of an organism's DNA that codes for a particular biological function. And the entire set of DNA that makes up a particular organism is usually the genome. Well, the sequence, DNA sequence, is just essentially a set of strings using the 4-letter DNA alphabet, the ACGT. Then what genome assembly itself is determining the order of nucleotides in the DNA molecule. So, like, as I'm going forward, if you have any question, you can unmute yourself and interrupt me so I can answer.
Sorry, Michael. Is there a way you could reduce the interference, because there's this background interference with your voice, like air, like wind blowing into your mic or something.
Is it better now?
So we can't hear you clearly. Yeah, it's better.
Okay, sorry. So yeah, should I do a recap or? Let me just.
I think it's a problem with your mouthpiece. Sorry.
Okay. Sorry. So
you don't have to bring it very close to your mouth. Just keep it far from your mouth and it'll be good.
Can you hear me now? Is it better now?
Yes, it is. It is.
Okay, good. So
Thank you.
I thought bringing it closer was going to be better. Okay, so I'm going to be talking about genome assembly, and like a metaphor about what genome assembly is. You have these DNA clones. That is, you have many copies of a book, and you're passing those copies of book through a particular shredder, and you want to take those shreds and arrange them piece by piece again to form the original book that you have. So what genome assembly is about is reconstructing the complete sequence of a DNA molecule from short sequence fragments, which are called reads. And here we have the DNA clones here, and we have the reads. And upon assembly, you have something like this. So, okay. So there are different approach for assembly, for genome assembly. So you have the reference assembly approach and de novo assembly approach. So for the reference assembly approach, typically you have like a sequence of similar genome. So you already have a reference genome already. So you're aligning your sequence reads to the reference genome and you assemble them that way. So this way, you're using the reference genome to guide your arrangement or the construction of the genome. But it can also mislead you, though the reference assembly approach is used basically in human genomics. So when it comes to de novo assembly, so de novo assembly means that you don't have any prior knowledge about the genome. So let's take, for example, COVID-19. So we had the SARS-CoV-2 virus, and we wanted to sequence the genome of that particular virus. There was no prior reference assembly. So to construct the genome of that particular virus, the approach you're going to use is the de novo assembly. So which means that it's only the supply bits that you use. And you have no prior information about that genome. We have no prior information about COVID before. So all we had to do was sequence what we have, then assemble them together de novo. So there are some basic terms we need to know about genome assembly. You need to understand that what assembler use, they use overlaps between those reads. So the read first produce what we call contigs. Then we later use this contigs to build scaffold. So now the regions between contig 1 and contig 2 is those stated as N here. They are ambiguous reads. And from the definition here, a contig is a sequence of maximal paths through the graph, which means that what? These are the overlapping reads that the assembler can bring together before it come across any other ambiguous reads again. So here we can have contig 1 reading A, C, T, G, reading those particular sequence reads without coming across any ambiguous reads. So here we're going to have like those paired reads. So we have contig 1, we have contig 2, and we have contig 3. So this particular contigs is then being used to build the scaffold. So the metrics that we use to show whether a particular assembly is okay is the number of contigs and scaffold. So fewer is better, 1 is ideal, which means that what, the lesser the number of contigs and scaffolds that you have upon assembling your genome, you have like a better assembly. Then we also have the contig size. So the maximum contig size, the average and the median, and also the M50 are metrics that are used to check if the assembly has been done properly. So how do you know the number of N's in your genome or the sequence? So we use the N50 metric. So the way to get the N50 metric is by there's a calculation for it. Then upon assembly, you get the N50 metric. So the N50 of the set of contigs is the size of the largest contig for which half of the total size is contained in that contig and those larger. What does this mean? It means that what you try to check, let's say for example, you have like 7 contigs, totally you have like 20 units of those. So there's a way you add those particular contigs together and you get your N50. So if the N50 is lesser than 50%, which means that what the assembly to some extent is better. And joining too much contigs together can increase the number of N50 that you have. And all these are like terms that you need to know if you want to really build like your own algorithm for assembly. So I'll just go straight to the genome assemblers that we have. So to assemble your genome, upon retrieving the sequence, maybe directly from your sequencer, then what you need to do is just to assemble the genome. But before you assemble the genome, you have to choose your assembler of choice. So there are some particular criteria that you use to choose assembler. So we have different types of assemblers. You have Spades, you have Velvet, you have HGAP, we have Falcon, we have Canoe, and all of them, they have different description, what they are used for. For Spades is good for small genome, single cell, and it can work with wide number of technologies. It works with Illumina, Solexa, Sanger, Iron Torrent, the PacBio, and Oxford Nanopore, along with it. You work with them. The Falcon, we only work with PacBio reads, and we have Canoe that work with just long reads, and we have the Trinity, which is to assemble maybe transcriptome assemblies also. Now is from like Illumina RNA sequence. So we have these different softwares that we use for assembly. But how do you get to choose your assembler? So there are a number of things you need to consider when choosing a genome assembler. You have to consider the properties of the genome, which is the genome size, number of repeats that are there, the heterozygosity, is it a polyploidy organism, and also the GC content also determines what assembler should you use. Then the process that you use to sequence, assuming to get like maybe more genomic DNA, you amplified the DNA that you have. Then that means you have to use a particular assembly tool that is designed to work with amplified DNA. So because if you use another tool, you're going to have misassemblies. So in the case of maybe whole genome amplification, Spades will work very well. And the sequencing technology used, that is like I told you, maybe did you use Illumina to sequence? Did you use PacBio, Nanopore, use Sanger, or use Iron Torrent, or the different sequence technology that we have? Which of them did you use to sequence your genome? So that will also determine the genome assembler that you're going to use. Then also, which is very, very important, computational resources. All of the computational resources, it's actually maybe the power, like the performance of your computing machine. So basically, they're analyzing or assembling your genome maybe on a server, AI performance computing machine. You might need to worry less about the computational resources. But if you're going to use your local PC, your computer, you have to be able to work with samples. Okay. 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 Okay, sorry about that. So the computational resources I was talking about depends on the performance of your computer. So the computational resources, maybe if you're using your local PC, you have to consider the number of like the RAMs that you have. If you're using an 8 gig RAM computer, you might want to choose your assembler carefully. Also, going from here, this is the basic genome assembly workflow. So you have your raw read sequence from the sequencer. So the first thing you do is check the quality. That is data inspection. So you check the quality. Oh, the quality is not good enough. Then it needs some, clean up. Then you use what you clean. Then you assemble with your parameter sets. Then is your assembly okay? If your assembly is okay, then you have your draft assembly. It's just kind of straightforward. And genome assembly is like the first step you do in any bioinformatics work. If you are working with raw reads coming from your sequencer, or the first thing you need to do is to assemble the genome together, because you have to put them together in the order in which like the original order, putting them like chromosome by chromosome, stuff like that. So that is actually what you need to do for genome assembly. So I'll go to annotation and importance. So really why you actually like assembling your genome together. I know you want to assign a possible function to a string of nucleotides, which means that what you want to arrange those nucleotides in a way that what they will match with a particular gene that you know. And once you know a particular gene, then a gene has a particular biological function. And actually, annotation is actually more than finding genes, as we know. It's very, very important for all the other stuff that we need to do. No, beyond genome annotation, you can get candidate genes, which means you can get new genes upon annotation. Then you can get similar known genes from other species. And by genome annotation, you can get targets for crop improvement, treatments for genetic diseases, comparative genomics, then study the evolution of species. Then what makes a particular species unique is also why you're actually annotating the genome, and also want to get if the particular individual is unique from the other individual that is actually getting variants. So an individual in a particular species, you want to check, oh, what variant does this particular genome has from the other one? Okay, so like I said, why do you also want to assemble an organism's DNA? You know, determining the DNA sequence is very useful in fundamental research into why and how a particular organism lives. And it is practically useful in any biological research, knowing, like having the knowledge of a DNA sequence of a particular organism or any living thing. You want to be sure that you have the correct sequence, which is why genome assembly is really like the first step like of the stuff you do. Once you have your genome, your raw reads coming from the sequencer, you don't have like a draft genome actually until you assemble your genome together. And you know, in medicine, it can be used to identify, diagnose, and potentially develop treatments for genetic diseases. And similarly, research into pathogens may lead to treatment for contagious diseases. So, okay. Hello.
Hi, Michael.
Yeah. Is there any question?
There's no question, but let me put it out. Please, if there are questions, please drop your questions in the chat session.
Or if you want me to do like a very quick recap of what I just said now, before I go to demonstrate the whole genome assembly pipeline. So,
okay.
Boss, sorry, I don't know if I'm supposed to use voice. I'm sorry to chip in. Please, I'm trying to make sense of how to link the first speaker and this second one. Is there a way we can understand, maybe after getting this sequence from the NCBI database, for example? Is that what you mean by when you say the fresh data, and then you clean it up? I didn't get that part.
Okay, thank you. So what Shane was talking to you about was about retrieving sequence from the NCBI database. So depending on the kind of sequence that you are retrieving, you might be retrieving raw reads. Raw reads means that it has not been worked upon at all. The way the reads is coming from the sequencer. So that is what raw reads about. So when I say fresh data, I'm talking about something that's coming straight from the sequencer, from whatever sequencing technology that you're using. So straight from the sequencer is the raw reads. Do you understand that?
Yes, I get it now. Thank you.
Okay. So any other question?
Okay. So someone also requested that you recap. You do a quick recap like you suggested.
Okay. So the recap is this is it. You upon let me just take it to this particular project you want to do. Let me take COVID for example. Now COVID is just coming for the first time in Nigeria. So the first patient or the first sample we were able to get for someone that was positive for COVID, we now want to sequence this particular COVID. Are you getting me? Sequence that particular pathogen. So upon getting your DNA from the person, whatever, maybe the saliva or the blood, then extract your DNA, then go through the molecular biology workflow, then use your sequencer, then you sequence the DNA of that particular SARS-CoV-2. So what you are getting directly from the sequencer is called raw reads. So those raw reads, you cannot work with them unless you reconstruct them. So that is why, like, that I was trying to like use this metaphoric illustration to explain what genome assembly is. So this DNA clones is what you are getting from that particular patient. Then upon doing all the molecular stuff and you sequence the genome, you are going to have these reads this way scattered. So you're passing the paper through a shredder. So many copies of the paper. Why is it many copies? Because you really want like the DNA, the genomic DNA might be rich. So it's not as if like it's just a particular, how do I put it? It's not just a particular copy. So there are different clones of the DNA that you are getting from that particular sample. Then upon sequencing it, you have different coverage, which means you can have like maybe times 40 coverage, means that what a particular part of the genome, you have covered that particular part of the genome 40 times. So what it means that what you now want to arrange those particular reads together and recover a constructed genome. So it's from that constructed genome that you can begin to do variant calling form, do alignment form. So because if you don't have that particular draft genome, you cannot do anything because you just have raw reads. So that is the essence of genome assembly. Actually, the first step. Once you get your raw reads from the sequencer, the next thing that you do is to reconstruct those particular reads together into one stuff. So that is why you have this recovered genome in this particular case, having like this particular newspaper. After shredding it, then you arrange them together. So looking at this particular one now, so this is like the original copy of the sequence in multiple copies. Then it goes through this particular sequencer. They have them in reads. So the reads are very short. Reads are very short and they may contain errors and biases. So that is why you need an assembler to reconstruct the original sequence from the reads. So that is why we're actually doing genome assembly. And I told you a different approach to it, the reference assembly approach or the de novo assembly. The reference assembly is like, you have something you're working with already. You have like let's see, human genomics now. If you are working with cancer, you don't need to say you might do de novo assembly, but like you have the human reference genome already, so you can align the particular that your raw reads then to construct. So that reference assembly helps you. It's like a guide. It's like maybe you're solving a puzzle. So we are trying to like arrange those puzzles together. You know the end point of where you're going to already. You know the end point of, so you just try to rearrange everything together. So de novo assemblies, you don't have any prior information about the genome. That's why I brought the COVID example. Like when we had the pandemic, nobody had the reference assembly or the reference genome sequence for COVID. So we had to do de novo assembly. So I just explained some metrics. So, and these metrics, we are going to understand it when I do the practical stuff. And then all these are just softwares that we use for genome assembly. Then these are criteria for picking genome assembly. I told you that what you have to understand if your laptop can do it. Did you use Illumina? Did you use PacBio? Or did you use NanoPore? Then the other one is doing your the extraction of DNA. Maybe your genomic DNA was very small, and you had to use whole genome amplification process before you can sequence it. You have to understand those particular stuff. That's why you have to, you can't just say you have to just be on your computer and do the bioinformatics stuff. You have to understand the biology of what you're actually doing. So that is why you have to understand that for the whole genome amplification, the genomic DNA was very small and it needs to be amplified. So for that, there's particular software that will help you to reconstruct the raw reads that are from that particular process. And you have to understand the genome you are studying. So if you're a parasitologist, you understand that the GC concept of maybe Plasmodium is iron. So all those stuff helps you to pick a genome assembler. So I just explained the workflow. So when you have the raw reads, the first stuff you do is data inspection. Then if the data is bad, you clean up or you trim. Then you assemble. So I'll just go straight to like showcasing the pipeline practice. So also I'm going to go to the terminal. Okay, so yes. Okay, so the first of actually is having your sequence in like wherever you are. You get me? Like where them coming. So okay, so sorry, I'm trying to log into where I'll do the practice for us. Okay. Okay, good. So for the assembly, like there are different ways you might want to like assemble different platforms. But because like normally the genomes, they are very, very large. So you want to make use of like the Linux-based software. You don't want to use maybe probably a web-based platform, maybe like Galaxy. So yeah, so you have to, what the first step you need to do actually is to get your sequence together. So for me, I have my sequence downloaded already. So I'm just going to list them and show you the way they are. So I'm going to list. So actually I have my sequence. So I have them in FASTQ format. So Shane was trying to tell us the different format of sequence used. We can have them in FASTA, have them in FASTQ. And we also have the other ones. We also have some like FAST5. Like we are working with maybe nanopod sequence. So for nanopod sequence, what will come out is going to be a FAST5. That is going to be like the format of that particular sequence. So now I have like this particular sequence. So it's a paired end sequence, which means that you have the forward and the reverse sequence. So it is named 1 and 2. So like I said, I've done the whole stuff, the whole molecular work, and from the sequencer, I have my readings already. So the first of all I'm supposed to do is to check the quality. And from that is from here, I said the first workflow. Let me go to workflow again. So you have your reads. So you do data inspection. You want to check if your reads are actually very good before you go ahead to assemble them together. So you assemble gates. So from here, I have my terminal. So a tool for checking for quality control is FastQC. It is FastQC that I use for quality control. So here I just do FastQC, then the file. So I'm going to do for the forward sequence. Okay. Okay. So this way is going to run. It's going to start the analysis. Though I've done it before, I just want to show you how this works. I'll do for one of them. I've done for all of them before. So I just show you. So this way, I'll use FastQC. So I might not be able to teach you guys how to like use the whole commands and everything, but I can share the pipeline like for what I've done. So this analysis is to check the quality, and from here. So what I said is we're checking the quality. So data inspection. So now we are done. So it's going to give me in a FASTQ format. So but actually I've done that before. So I have the FASTQ report somewhere here. So now, this is the FASTQ report. So I'm checking the sequence quality now. So now, like basically, all you need to do is this first graph is what you use to check if the quality are good. Now, if you check this, once to know that if your sequence is bad, once you have this bar touching this red zone, actually, basically like the first of your sequence is bad and it needs cleanup. So here, this particular graph is showing that the per base sequence quality is showing that this particular sequence is bad. Like I did this for the reverse sequence too, and it was the same thing. So now, if the other stuff, the GC content seems to be, I think the N content, N is the ambiguous sequence. What I mean by ambiguous sequence is those sequences that the sequencer cannot like assign nucleotides to. So they are not A, C, T, or G. They are not any of those nucleotides. So you just give them N. So check this. So the per base N content is actually okay. But here, where we check the base sequence quality, and this is usually in Fred score, and this is actually the positioning of the read in base pair is actually bad. So you don't want this bar to be touching this red part. Actually, you want it to be in this green part. This green part means it's actually safe. So the tool for cleaning that we use is, or trimming, is a Trimomatic. We have other tools too. We have Cut Adapt, we have FastP, and we have several tools for cleaning your sequence. So what actually you want to clean from the sequence is the adapter sequence. So the adapter sequence is what was used during the sequencing. So you want to cut off those adapter sequences because they might be affecting the quality. So after that, after you are done with the quality control, I'll show you the commands that I use. So I don't want to run it again. It's going to take a while. I'm just going to show you the command that I use. So upon trimming this file, what you do again is what? You go back again and check the quality because you want to be sure that what you've trimmed is actually fine. So, and this is the FASTQ report of the trimmed format. So now you can see that what I was talking about, the per base sequence quality. Now you can see that what all these bars, they are not touching the red or this particular, they are in the green region now. So which means that what your sequence is actually good now. So you can go ahead and assemble your genome. So the step is basically simple. Check the quality. If the quality is bad, you trim. After trimming, you check the quality again. So if the quality is good, you go ahead and assemble your genome. So right now, I can say that the sequence is good and I can go ahead and assemble the genome. So for me, I used spades for my assembly. So there are a bit of commands that you type, but I know we're not really I can't really type in those commands and everybody see because it take a while, basically. So upon assembling your genome, let me see if I can get I should get the command. Okay. Sorry, I'm trying to get the command that I used so I can. Okay. So all you need to do, the command is usually spades, because spade is written in Python. So you use spades.py, then flag one means the forward sequence. So you can see that I've renamed it as the trimmed version, because you're working with the trimmed version and you work with the original rubies that was getting after you've trimmed it. So you work with those ones. Then flag 2 means what? The reverse sequence. Then what? Flag O means the output of where you want your assembly reports to be. Then after you run this, you should have something similar to this. I'm coming. You should have something in this folder, spades assembly. So in spade assembly, you have different stuff that you need to work with. So now upon assembly, you have to also check the quality of your assembly. So for every analysis you do, most of the time you have to check the quality. So assembly with parameter sets. After choosing the appropriate parameter set that you want, then there's this particular person assembly. Is it okay? If it is not okay, you have to go back again and reassemble the genome. So I have to check if the assembly is actually okay. So the tool that we use for this is Quast. So Quast is a quality assessment tool. It's used for checking the quality of assembly. So once you run the Quast, because I will not run the Quast here because of time also. But once you run the Quast, you're supposed to get a report, also like HTML. So you have your Quast report written this way. So because I did this today, so it was 15th of June. I did around this evening around 7. So the Quast is quality assessment tool for genome assemblies by these people. So now all you have is this particular graph, and you have some statistics here to show you if your contigs and scaffolds are actually okay. So right now, what you're actually most concerned about is the scaffolds, because like I told you, you get the scaffolds from the contigs. So there's an heat map here to show you if it is worst, if it is actually average, or if it is the best. So checking here, so you can see the heat map here showing that what for the total length, when it is greater than 50,000 base pair, you have actually very good scaffold according to the stuff. So you also have to check the GC content. Is it actually okay? You have to check the M50. So I see the M50 here is actually okay. So reading from here, the report is actually very good, because if you don't understand anything, it shows you. If you click on M50, it tells that M50 is the content length, such that no longer stuff. It shows you actually what it means. Shows you the L50, shows you the L90, shows you the GC content and everything. Shows you the number of mismatches, what they cannot be construct together. So right now, I have a lot of assembly is good, but no, most of the time, after you are done like with this assembly and you have a draft of the genome. So those drafts are really going to be in the assembly folder. So here you might have it in the scaffold.faster. So you have it here. So this is probably like your draft, the draft sequence that you have. Now, I was talking about annotation. So imagine like you just collected the sample from somebody, you did all the process. You want to be sure that I have sequenced the right organism. So that's when you now do annotation. A bit of annotation is trying to check what organism it is before using some other tools for annotation too. So I can open the scaffold.faster and blast it in SCBI to check which of the organisms or which particular organism is this sequence for, because we're working with a particular sequence, and so far I've not told you guys what the sequence is. So if the assembly is actually very okay and you know what the sequence, you'll be able to blast it and say, oh, this is what I've done. So I can do not scaffold. So I'm trying to view, so I can copy just some part of the sequence of this particular draft assembly, then try to blast it on NCBI to check which organism does this particular sequence. So I think I just copy. I can copy this whole first stuff. So, sorry, trying to. Copy it. So I can come to NCBI. NCBI. Let's search for blast. So what blast is a basic local alignment search tool. So it's what it does, it's trying to check for local similarity between sequence. So because what I'm looking for is not new, are you getting me? I believe that the sequence is already in this particular database already. So it's not de novo sequence. So I can say that, let me blast and check even though it is de novo, I want to check, is de novo COVID, what organism is the closest to this particular stuff that I've done. So I can do what I'm going to do. I'm going to do blast, nucleotide blast, that is nucleotide to nucleotide blast. So there are different type of alignment, and I'm sure in the next session, Adido is going to take us through alignment. So probably have to do this one too, but I'll just go straight to using the nucleotide to nucleotide alignment. So I want to check which other nucleotide is very similar to this. So I can just paste the faster sequence I copy, I paste it here, then I can just, it's just one I want to align. So if you align more than two sequences, you click here, then just go with the defaults, then just blast. Then you're looking for highly similar sequence, run for some time. So I don't know. I don't know. It might take a while, but why it is running? Okay, it's out. It's out. Okay, now. So you can see that I have the result already. And the organism I'm going to get is the Escherichia coli, this particular strain. And so what I actually assembled together was Escherichia coli and the strain of this particular Escherichia coli. And I might try to check how similar it is. So the query covered was 100%. the percentage identity was also 100%. So which means that what actually sequenced this particular, you can see 0157 dot dot H7. So there's a particular string of equivalent that I've sequenced. But assuming I copy the O fastest or the O file, and I might get the exact sequences, but I have different, there are different strain of this particular Escherichia coli itself. So what I've done here is to check, is my assembly also correct too? So basically, the essence of assembly, like I said, is the first step before any genomics pipeline. Once you have your raw reads from your sequencer, genome assembly is what you do. So I will take questions now. So, let me see. Is there any question in the chat box? I can take question. Okay. Is there any question? You can type it in the chat box. I'll just be waiting for you guys. You can unmute yourself and also ask your question. Okay. How can we learn these commands? So I used like a Linux-based OS to assemble the genome. We can also use like Galaxy, but then to learn this command, all you need to learn is just learn the basic Linux commands, like moving from directory to directories, and use them over time. I guess you mean. So you have to learn the commands on your own. There are YouTube resources I can share on the group chats. There are courses you can take just to learn the Linux commands. But basically, you have to learn them on your own. And it's actually, once you begin to use them, you get familiar with them. So any other question? Somebody said, I think you should share your slide afterwards because I'm lost. Okay. Okay. Esther, can you tell me where you got lost? Like maybe I can do a recap because I think I still have some minutes. This is just like I was trying to rush everything so I can like show you guys the whole pipeline stuff, the way it works. But basically, the stuff that you don't understand, if it is the commands, I might not be able to explain the commands. But if it's the concepts that you need to understand, I'm able to explain the concept to you, Esther. Yes. So Daniel Adediro. So the assembly I did is de novo. I get you. So there was no reference at all. Yes, you are right. It's de novo because I was not using any reference. So I'm just assuming that what organism is this? And I just tried to do de novo. What I did was de novo. Okay. So now reading. Saying for people using Microsoft OS, how can we get the terminal using CG Web and Blink? I don't know what CG Web, but then every terminal can use Blink. Blink is a Linux-based software. So if you're using Microsoft OS, we start trying to like install a Linux operating system on maybe on your laptop. You can also use cloud platform like I think AWS, there's Google Cloud. So I might share, I can share these resources later. How you can sign up to use Google Cloud platform. So the Google Cloud platform has a Linux based like an Ubuntu OS system that you can use there. And the AWS also have. They also have I think the Cloud Shell or Google Cloud Shell. So also it's also a terminal that you can use too. Okay. So any other question? You can unmute and ask also if you
please. I want to finally get this thing. Some of us are practically JJC in this stuff. So
Okay.
whatever I'm going to ask, please don't make it seem as if maybe what this one's saying.
No, it's
I'm
all
just
right.
trying
It's
to
all right.
put everything together. Okay. From what you said so far now, correct me if what I'm saying is wrong or right. Say there's an organism
Mm,
and I want to determine which organism it is. I don't know. I take the organism to the lab, Abi. I sequence the genome, or sequence a part of the DNA. After sequencing it, then we'll pass it through all this process you just mentioned. Like identify if the sequence is good. If it's not good, you go back again and piece them together. When you're done, you just blast.
yeah. Okay. So yeah, so basically
Sorry.
you're correct. You're correct.
Mm.
But the stuff is it's not as if maybe you don't really know the organism that you want to sequence, I guess. That you're actually sequencing. So in that case that you don't know the organism you're sequencing, that one is metagenomics. You're sequencing a lot of things together. But this one, in the process of sequencing this stuff, there's in the molecular biology aspect, you have done a lot of things to make sure that what we come out of that particular sequencer is going to be the organism's DNA, either by amplifying the DNA of the organism during the molecular biology aspect, or maybe a way to just make sure you get, if it's histology, make sure you get the parasite DNA out of it, then you are sequencing the parasite DNA. So what the de novo assembly is that because you don't want to, because the reference assembly approach might be misleading, and you don't want to maybe miss out new variants. So in terms of you're trying to determine maybe new variants, new anti-malaria variants in Plasmodium falciparum, you don't want to use reference assembly because it might mislead you to miss that particular variant you're looking for, maybe novel anti-malaria variant, drugs variant. So you want to use the de novo assembly to reconstruct the whole genome the way it is. Then after that, you can begin to do your variant calling and any other bioinformatics work that you want to do. So I think that is it. Do you have any other question, Idris?
Thank you very much. I'm grateful.
Okay. Okay, so do I need a very strong programming background in order to survive? Sorry, Emmanuel. Hello.
No, I was calling your attention to that question that you are about to attend to now.
Oh, okay, so do I need a very strong programming background in order to survive, or just basic knowledge is fine? So the stuff is like, you don't just like have strong programming background. You have to start from somewhere. So the basic knowledge that you have in programming, because basically I've not really done any programming. What I've just done is just type commands. So I've not really done any programming at all today. So you can learn. So depending on what you want to do, if you want to do genome assembly and you want to, just all you need to do is just probably using a Linux system for a while, and you want to register for maybe a Python course, it's not going to be relevant because you're trying to think what you need to do is learn programming. But what you need to do to just achieve that particular task, to solve that particular task, is learning Linux command. So in this particular session that I've taught you guys, I didn't use anything about any program, any Python or R. In fact, even the commands that I was using, I didn't use any bash scripting, maybe writing some maybe loops or something to make the stuff faster. I type them one by one. So it can that way it's simpler. So you don't need a very strong programming to get started in bioinformatics. But in order to survive, maybe going to other aspect of bioinformatics, maybe the data science aspect, maybe after this genome assembly, you now want to probably plot the assembly report yourself in a better way. In that particular instance, you might need the knowledge of R or Python to do a bit of data science. Like you need to first get one very strong programming background before you can get started in bioinformatics. With the little knowledge you have, any little knowledge you have in what you need to do. So I'm not saying you don't need to learn Python. For what you need to do, what I've like taught you guys today, I didn't use Python or R. What I just used was the Linux command line and Linux commands. So that's it. I don't know if there's any other question or not. So. Okay, yeah, thanks.
Yeah, good evening, Michael.
Okay.
Yeah,
Good evening.
good evening. Thank you for the wonderful presentation. So I think I've had an interesting learning opportunity so far from Sean's session and your session too as well. But now I'm just like thinking out loud, right, based on this field of bioinformatics and, the applications that it has, in the world at large and its futuristic opportunities that we have there. So what applications? I just want to really understand that. Do people use bioinformaticians apply these skills to, what do you use it to get? Is it just basically just sequencing genomes and maybe finding out new variants and all of that? So I just want to get the application of this so I can really just put some thoughts together. I still have other questions, but I want you to answer this first.
Okay, so if I what are the applications of these mathematics skills? Is that right?
Yes.
Okay, good. Okay, so like you said, is a skill. Is a skill for every biomedical researcher. So if you are doing something in the biomedical world, or basically if you're a life scientist, you need a bioinformatics skill to survive in this emerging world right now. Why? Because everything that has to do with molecular biology has expanded from molecular biology itself to genomics and or transcriptomics and the rest. And what bioinformatics is just trying to help you make sense of what you have. So, you want to why do you sequence after the whole explanation I said about the importance of genome sequence and everything. So you want to make sense of the genome of the sequence that you have from the sequencer. So anybody can operate this the sequencer. They can get the technician that has worked with maybe Lumina before and does not have any knowledge about bioinformatics at all. Then the person just needs to do is just go to the lab, extract DNA, do all the whole workflow for you, then get you a sequence a skill format. Then from there, what will help you to make sense of the sequence that you have is your bioinformatics. There are different approach, different application rather of these bioinformatics skills. There are different ways you can make sense of a sequence. And so that is why we have different application of bioinformatics in precision medicine, in variant calling, checking for antibiotic resistance, anti-malaria resistance. All this stuff are all linked together but there's a general beginning to everything. You have to have a sequence first before you can say you want to do bioinformatics. But the other part of bioinformatics too. They are different. You might sequence the transcriptome. My sequence might be a single cell sequencing. So there are different ways. So you can apply these bioinformatics skills. So in single cell sequencing now, assembly those stuff together will be different than what I just explained to you today now. If it's a transcriptome sequence, it will be different. So, but the important stuff is the question you're trying to answer will determine what you want to learn, what you want to do, how do you apply this bioinformatics skill. If you're trying to answer evolution question, then you begin to see that, what I need to do is maybe you want to understand epigenetics, for example. You want to understand DNA methylation, how are genes turned on and turned off. You want to use the sequence that you've gotten from the sequencer to understand this. The approach will be used to check, how many genes have been methylated, how many genes have been unmethylated, how many genes have been demethylated, stuff like that. Or you want to check maybe histone and chromatin modifications in the sequence to answer a particular question. The particular approach is going to be different, but all of them boils down to the fact that you have a particular sequence that is coming out from a sequencer. So I I hope I was able to answer your question, Lighton.
Yeah, I think that is really profound. Thank you. I'm glad I asked, and I like the depth that you put into it. So, do you do some kind of like extraction work, like personally? Do you have any idea of DNA extraction, purification, and getting the maybe to work with a machine that can get the genome for you?
Okay.
Do anything like that?
Okay. So are you asking me personally?
Yes, I'm asking you personally as a
Okay. So personally, I don't really do much of like molecular biology, but I have idea, like I have experience in DNA extraction and stuff like that, and some basic molecular biology skills, maybe in getting parasite, because I'm like my background is in parasitology, so maybe in getting parasite DNA out, maybe get a blood sample from human, and don't want the human genomic DNA contamination. You just want the parasite DNA out to just amplify that.
Okay.
Deplete the leukocytes. So I have those basic molecular.
That's fair. Yeah, I just wanted to get that clear. So I was going to ask another question, but I just lost my line of thought. But I'll try to remember. So basically, your work starts from getting the genome. So then you now use several bioinformatic tools to answer whatever question you're trying to.
Exactly.
Okay. All right. Thank you kindly. Yeah. Thank you.
Thank you.
Yeah. So if I would ask, you use different tools like programming to answer. So after the genome has been gotten, you use what tools do you use?
So like I said, depending on what you want to answer. I said I used to right after assemble. I think first off after you get your genome is to assemble. So that one is standard everywhere. So you assemble the genome. You have to reconstruct it to the exact format in which it's supposed to be. Then let's say, what I want to check is what are the variants responsible for maybe albinism or something, or the mutation, or I'm just trying to check a trait that maybe, so you have your the sequence of maybe an albino person. You want to check what are the mutations. Maybe you know already, you want to check, maybe there's some other extra mutation that are available. Then that particular, what you do is what you do variance coding. You begin to reference that particular genome draft that you've gotten from the albino person to maybe a normal person, and try to check what is the difference between these two persons. Or you can also do all those case study procedure to a disease person and a normal person, try to check from the sequence of a normal person and the sequence of a disease person. I want to compare both of them. Where are the variants in the normal and the sick person? So what I've mentioned, that they require different tools, and depend. So the tools are open source. Almost all the bioinformatics tools are open source. You don't pay for any of them because they are happy that you're able to use them and cite them in your research. So they are open source. You just have to go read the documentation, answer them. And the basic stuff is that once you can use a particular bioinformatics tool yourself and reading the documentation, going forward, you should be able to use any bioinformatics tool. You might have some hiccups, but then once you understand the basic stuff, you should be able to use any bioinformatics tool.
Nice. You've answered my question. Thank you kindly. Yeah.
You're welcome.
Yeah.
Okay, so someone is asking, is there any bioinformatics books that I can recommend? Well, for books, they are textbooks. If you just search on the textbooks, but basically for myself, I've not used any bioinformatics textbook before. So it has just been working on projects, taking courses, and all. But then there are textbook, free PDFs and textbook online. But because I've not used anyone myself, I'll not be able to recommend.
And I'm sure everyone actually got something. And I understand Idris' position here. I was almost, I will not lie, I'm in that place. Even though I've taken some, I've gone through some materials before now about bioinformatics, but it's still definitely is as confusing as it has always been. So the request has been made before up until now. Please, we would like to go through your slides after this presentation, so for us to for us to check on some things, both for you and also for Sean.
Okay, yeah, that's fine. I'll do that immediately.
Thank you. Okay, so thank you very much for your presentation, Michael. Michael, it was a wonderful session,
Yeah,
and we
thank
appreciate
you.
contribution tonight. So hello everyone. Thank you for waiting up until now. I noticed the number of participants when we started, and I noticed the number of participants we have now. And I would say it's actually a good number starting, and it's actually a good number presently. And so we've all really given ourselves to this, and I appreciate everyone present and everyone that stayed up until now. So I would say that will be all for today's presentation. Let's look forward to a further application of all we've learned today. A further application will be made on Saturday. We have one more speaker one more speaker or instructor that is yet to speak to us on this area of bioinformatics and its application. That is are they doing this way, and we'll be having her speak to us on Saturday. But then, so far, how has the session been? I just want to hear one or two before we move forward. Before further information are passed across. Please, how has the session been so far? How has this session been so far? Please just drop some comments, drop a response. I would like to know how we've enjoyed the session so far. Hello, can you hear me? Can you all hear me? Hope you can hear me.
Yes, we can hear you, Emmanuel. It has been a wonderful session so far.
Thank you very much. Any other person? Okay, anyways, it's been a very wonderful session. I wouldn't like to keep us on for too long. Thank you for as many of you that stayed up until now. As interesting as this session was, I'm very certain that Saturday's session will be far interesting because we would get more information on this topic. So what I would employ every one of us to do now is to go back after tonight's session and just do a little bit of studies or studying on these areas that have been mentioned and how it can be applied in our various areas. Because remember the assignments Shehu gave to us. So the area of application, how it applies to that your area of interest in science, and then just now prepare yourself fully for that Saturday session. So thank you very much, everyone. It has been a very great night with us. Thank you very much, Sean. Thank you very much, Michael. And we look forward to another amazing session on Saturday. So good night from my end. I noticed we have a lot of discussions on this. Please let us all keep our questions intact. You can bring your discussion to the chat box on WhatsApp or save your questions till Saturday and then just drop it out. Thank you very much. Good night.
Good night. Good night, everyone.
Yeah.